All right. Thank you so much for joining us for this meeting. We have a very interesting topic to discuss today, and our presenter is Dr. Karen Dannemiller. She studies the indoor microbiome and indoor chemical exposures in relation to childhood asthma. She graduated with honors in chemical and biochemical engineering from Brown University and earned her PhD at Yale University in chemical and environmental engineering. During this time, she completed an internship at the California Department of Public Health in the Indoor Air Quality Program. She is now an assistant professor at Ohio State University with a joint appointment in civil, environmental, and geodetic engineering and environmental health sciences. In 2017, she was awarded the Denman Distinguished Research Mentor Award. At Ohio State, she leads the Indoor Environmental Quality Group. Okay, then Karen, I give the microphone to you and go ahead and start it. Thank you.
Great. Thank you so much for having me and for the nice introduction. I'm really excited to be here today. And my background is really largely in environmental engineering, environmental and chemical engineering. And I also work on public health concepts. So I really like working on interdisciplinary projects. And I'm excited to talk with everyone here today who's from the healthcare field about some of the different measurement methods that we can use and some of the projects that we're working on developing to produce better measurement methods to understand exposures in the indoor environment. So can you go to the next slide, please? Thank you. So I work primarily on childhood asthma with my disease outcome of interest. And asthma has both environmental and genetic causes. And we have a lot of tools in our toolkit to treat to treat asthma from a medical perspective in terms of medication and things that we can do. But I'm really focused on the environmental issues surrounding asthma. And we know that environmental exposures are an important component of asthma treatment. And one thing that we can do to better understand the environment is to improve our measurement methods of what we can measure in the indoor environment to both find better associations between these exposures and health outcomes, and then eventually be able to remediate those remediate those exposures to improve health outcomes. So today I'm really excited to talk to you about two potential environmental triggers for asthma. The first is formaldehyde and the second is mold. So as I discuss both of these triggers, I'm going to describe what we're doing now in terms of improving these measurement techniques. So first I'm going to talk about formaldehyde. So we have a long history of formaldehyde exposures. It starts back in the late 1800s when people would actually put formaldehyde in milk to make it last longer. And it would indeed preserve the milk, but it did result in harm for a lot of people, unfortunately. In the 1950s, there was a lot of formaldehyde present in textiles. In the 1970s, urea formaldehyde foam insulation was installed in homes and resulted in high levels of formaldehyde exposure. In 2005, after Hurricane Katrina, there were a lot of trailers provided by FEMA that were contaminated with high levels of formaldehyde. And more recently, in 2015, there were suboptimal manufacturing practices that resulted in flooring from lumber liquidators that was contaminated by a high level of formaldehyde. So all of this has resulted in a long history of formaldehyde exposure, but we don't yet have a great way to actually measure formaldehyde in the indoor environment. Next slide, please. So why do we care about formaldehyde exposure? Well, it's ubiquitous in the indoor environment. It's pretty much everywhere around us. It comes from pressed wood products like plywood and particle board, adhesives, paints, and a lot of other common indoor materials. And in terms of health effects, it's an eye, nose, and throat irritant, and it's also important for children with asthma. It's also a known human carcinogen, so it has a lot of important health effects, and there's a lot of reasons why we might want to understand what our formaldehyde exposure actually is, so that we can actually think about potentially reducing it if it's too high. Next slide, please. So formaldehyde, in addition to having these important health associations and being everywhere in the indoor environment, is also notoriously difficult to measure. Right now, the gold standard measurement technique for formaldehyde in the indoor environment uses these DNPH cartridges, and the cartridges are very accurate, but they're very difficult to use. So you need expensive analytical equipment. You also need a trained technician who knows how to run these samples, and it can be a really difficult process. There are other techniques available, but they're usually some combination of difficult, expensive, time-consuming. They're often prone to contamination because formaldehyde is so ubiquitous. And so right now, there's no widely used, accessible method for the average citizen or concerned residents, or maybe public health nurses who are going into homes and maybe want to measure formaldehyde to actually conduct this measurement. And so this is what we set out to develop, was a better formaldehyde measurement system that could be used more for the general public, as well as perhaps public health nurses in the future. So next slide. So what we developed is we actually developed a smartphone-based tool to improve formaldehyde measurement in the indoor environment. And we wanted this to be pretty straightforward to use so that people could use it to get a better understanding of what their specific formaldehyde exposure is. So this project we called SmartForm. It stands for Smartphone App for Residential Testing of Formaldehyde, and it is published. You can find the paper in the journal Building an Environment. You can see it at the bottom. And overall, what this project was set to do was to pair a smartphone app with color-changing badges. And on the badge, you could see them pictured here. We have a calibration patch, which doesn't change color, and another portion of the badge that does change color when exposed to formaldehyde. And we use the app to take a before and after picture of the badges to know what the formaldehyde level is in your home. So you would take a badge, take it out of its package, set it out, take a before picture, wait 72 hours, and then come back and take an after picture, and the app can tell you what the formaldehyde level is in that environment. These badges are produced by Morphix Technologies, our industry partner for this project. So next slide, please. So an overview of the badge, we used a collaborative development model. So we wanted to work with citizen scientists and other people who would be interested in eventually using this product at the end when it was finally developed. So we wanted to have a really straightforward user interface, and that was definitely easier said than done. So we put a lot of effort into designing the user interface in a way to be user friendly. You can see an overview of that on this slide. And we also wanted to use the smartphone as a great opportunity for user education. So when people use this app, it can provide a lot of important information to them about the formaldehyde level in their home and what that actually means. Next slide, please. So as we develop this user interface, it consists of several different screens. 3 of them are shown here. On the first screen, when you first open up the app, it shows a button where you can choose to take a new test. And when you open up this test, you can see on the next screen over here, you could give it a title, and you can tap in the one area to take a before image. And what that does is it's going to do a check on the badge. I'll make sure it's okay and talk a little bit more about that later. And it's also going to start the timer. And once that timer starts, you leave the badge in an area that you're interested in measuring for 72 hours. And at that point, you'll get a notification on your phone that it's time to take the second picture. You go back and take the second image. Your smartphone will process the 2 images and then tell you what the reading is. We've also integrated some surveys into the app so that people can upload their data if they're interested, so we can create a database of formaldehyde readings, along with other survey information such as environmental characteristics, questions like if you have your windows open or if presswood products like plywood and particle board are present, things like that, so we could get some better information about the survey. Next slide, please. So once we created this interface, we also wanted to make sure that we had the badges appropriately calibrated. So we performed several different calibration tests to test the badges, including testing different co-contaminants. So those are other chemicals that could possibly react with the badge that are not formaldehyde but could be present at the same time. And we wanted to make sure that acetaldehyde, which is very similar to formaldehyde, as well as other VOCs. Here we were using a VOC mix from plywood and particle board to make sure that there was going to be no interference from those chemicals. So we did 6 different tests. We did a high concentration test, a medium concentration test, and a low concentration test. And then we did an acetaldehyde co-exposure test, a VOC co-exposure test where we use that mixture of VOCs from a presswood product. And we also tested the badge at high relative humidity to see if that humidity would interfere with the badge and the measurement process. Next slide. So we were very excited with the system performance of these badges. You could see the graphs over there on the right. Overall, we found that the badge could measure within a range of 20 to 120 ppb, which is a pretty good range for measuring formaldehyde in the indoor environment. The standard deviation of the readings was 10.9 ppb, and we didn't find statistically significant interference from co-exposure to acetaldehyde or a VOC mixture. We could also look at that potentially a little bit more. One of the p-values was fairly close. So we want to continue to look into that to make sure there was no interference, but we didn't see any large interference from any of those co-exposures. So overall, we were really excited about the system performance. We found that this technique could measure VOCs in the indoor environment in the range that we were interested in. Next slide. So another thing that we were concerned about in developing of this new measurement method was testing different lighting conditions. So if you're taking a picture of a badge and in a home with a lot of bright lights versus a home with not many lights, we wanted to know if that could interfere with finding the appropriate test results. And we did find that lighting needs to be within certain parameters. But fortunately, we can detect this using the smartphone and issue a warning to the user if the lighting is not within acceptable levels. And so you can get a warning on your phone that says, you don't have enough light in the room when you took the image. Please retake the image in a higher lighting condition or a lower lighting condition or whatever it needs to be. Next slide. We also integrated several other warnings into the system. So we found that, for instance, overexposure, underexposure for lighting, we could measure that with a smartphone, issue those warnings I talked about. We can also test for badge contamination in the before image. So if we found that the badge you're starting with was already contaminated, we can issue a warning for that. When there is high relative humidity in the room, the badge actually turns this kind of like blue-purple color, which is really convenient actually for use with the smartphone because we can also issue a warning if it looks like your relative humidity is too high and your test result is not going to be accurate. You will get a warning to retest. If the exposure time was too short or if you're below or above the detection limit, you can also get a warning. So we were really excited about this system in terms of integrating the smartphone and actually using a lot of features of the phone itself to guide users to using the system in the correct way where they're going to get the most accurate results. Next slide. We also did a beta test and a survey of some beta users because we wanted to know that the system was easy to use. And so we asked a series of questions in terms of challenges that people might have faced. And overall, it seemed that people found the system easy to use. They were pretty happy with it. It had a user-friendly design, especially after we did some reconfigurations based on some other feedback that we had gotten on it. The biggest challenge seemed to be actually accessing the survey and finding the right place to put that survey button so that people could upload their data if they were interested. And so we've reconfigured that based on the results of the beta test. So it's now a little bit easier to access the survey. Next slide. So overall, we were able to design a smartphone-based system for formaldehyde measurement. And we see this as a great launching point to use smartphones for detection of a lot of indoor contaminants that we might be concerned about. So overall, the takeaways were sort of, first of all, that this worked. We were able to do it and create a system where you could use a smartphone to measure indoor contaminants. The second main point is that this is going to be used as a screening tool. So this type of analysis is not going to be as accurate as that gold standard DNPH method. We did have a standard deviation of 10.9, where the standard deviation of the DNPH method is much, or orders of magnitude less. So it's not going to replace the DNPH method if you need a really accurate reading, but it's very useful in terms of being used as a screening tool if you just need to know sort of ballpark what your formaldehyde level is and you might want to reduce that level. This is going to be a good indication of if you have sort of a high formaldehyde level. In terms of future work for improving this system, we want to improve the risk communication. So we have some user education information included in the app if people are concerned about their formaldehyde exposures and what they might be able to do to reduce it. And future work could also improve precision. So we, could think about if we can possibly improve that standard deviation. And we also need to continue to work on better integration of the survey into the app so that people have better access to that survey if they want to upload their data. But overall, we are really excited about this project, and we think it has a lot of potential future applications for measuring other contaminants that you might be concerned about as well, using smartphone technologies that we now have available. Next slide, please. We expect that this will be available on Amazon eventually. We're currently working with the manufacturer now to get this out. You can download the app now if you want to check it out. You can just search for SmartForm, or you can also scan this QR code. It'll take you to the link, and you can go ahead and download the app from iOS and Android devices. And eventually this will be launched on Amazon, and you could purchase 5 tests for $25. And the nice thing about having multiple tests is you could test in either test in a couple different areas, or if you get a high reading, you could work to reduce the levels in that area and then see if it helped and your reading went down. Next slide. So overall, we're not sure what kind of formaldehyde exposures will happen in the future. Given the history of formaldehyde exposures, there very well could be other big events in the future where people are concerned about formaldehyde. And even if there's not large events, we know that formaldehyde exposure in the indoor environment is pretty ubiquitous, and some people may have high exposures. So hopefully we can help identify what those exposures are. Next slide. So hopefully we can stop a lot of these exposures, and having more accessible measurements might increase accountability so that people can know what their formaldehyde exposure is and what to do about it. Next slide. So overall, we developed a new accessible screening tool to measure formaldehyde, and we do have future work that we could think about in terms of improving precision as well as potentially making the survey access even easier. But I think this is an exciting new area of interest where we can expand contaminant measurement potential in the future to a lot of other different compounds that people might be worried about being exposed to in the indoor environment. Next slide. So obviously I did not do this alone. I worked with a fantastic team. They were absolutely wonderful. The team is listed on this slide here, including Dr. Kastner, who is a co-chair for this webinar. So I was really excited to work with everybody on the team. This project was funded by the National Science Foundation, and we also had a lot of help from a lot of community scientists who contributed their time to looking at the app, beta testing the app, providing feedback, as well as our industry partner. And we also got some design advice and worked with a community leader on this project. So I was really thankful to work with such a great team on this project. All right, so that sort of ends my section on formaldehyde and some of the measurement techniques that we're using to measure formaldehyde in the indoor environment and some of these new techniques using smartphones. Part 2 is going to focus on mold exposure in the indoor environment as well as exposures to different microbes. So I started out in chemical engineering during my PhD. I kind of moved over into doing some environmental microbiology, and now I do a little bit of a mix of both, of the indoor chemistry and the indoor microbiology. And you'll see that through my talk. But so I'm really interested in indoor microbial exposures because it's such an important problem, especially for asthmatics. So exposure to mold in homes costs an estimated $22.4 billion per year and contributes about 16% of the cost to allergic rhinitis, acute bronchitis, as well as asthma. So it's a really important problem. And it's usually not as pretty as this nice piece of artwork shown on this slide, but can cause a lot of really important health effects. But it's actually really difficult to find a good way to measure mold in the indoor environment because the exact causal mechanism is still unclear in terms of how the mold exposure is linked directly to these health outcomes. Next slide. So one important source of exposure, though, to fungi in the indoor environment is actually dust. And so a lot of the work today that I'm going to talk about focuses on measuring the microbes that are present in dust. And that's because anytime you walk into a room, you're going to kick up that particulate matter off the floor. And in an occupied space, about 83% of airborne bacteria and 66% of airborne fungi comes from that resuspension in the floor. And in fact, you can actually calculate microbial emission rates from people. So wherever you are right now, if you're by yourself in a room or if you're in a room with other people, you could actually calculate out what those human microbial emission rates would be. And a lot of that is actually going to be from resuspension of the floor, and some of that's going to depend on activity level too. So if you're running around the room, you're going to see more resuspension, or if you're just sitting still, there's not going to be as much resuspension from the floor. And so we've seen this around the world. So you can see a study here that looked at different places around the world at resuspension of these microbes. And so it's a really important source of our exposure is from the dust. Next slide. So we want to work to improve measurement of these microbial exposures from the dust. And one reason that it's been so difficult to measure these microbes from the dust is because previously the only way that we had to do it was using culture-based methods before the advent of a lot of these DNA-based methods. So if you were using a culture-based method, you might set out a petri dish like this, and you could have different types of fungal fragments that land on it in a room. So you could have a viable spore, a dead spore, spores that can't grow on the media that you used, an unidentifiable spore, and other fungal fragments. And all of these can potentially contain allergens and have potential health effects, but only some of them are actually going to grow into colonies, as indicated here by the viable spore and the unidentifiable spore. And then only some of those colonies are actually going to be able to be identified. And in fact, only about 17% of fungal species are culturable at all. So when you use these culture-based methods that were used extensively in the past, because it was all we had for these environmental analysis, you're just getting such a limited picture of what's actually there. And you're generally going to see what grows fast and what likes the type of media that was used on these plates. Next slide. So fortunately now we have a lot of DNA-based methods where we can measure all of the different microbial components that are present. So you could collect the same types of microbial components on, say, a filter, and you could extract the microbial DNA from all these different components, and then you could read the DNA just like a barcode to tell which specific fungal species that DNA came from. And this allows us to get a really comprehensive picture of the microbial exposures that you're having in the indoor environment. So you can think of an analogy of this of all of the research that we've been seeing related to the human gut microbiome. And we're applying a lot of the same methods here to understanding the environmental exposures that we have every day in our indoor environments, where we can now get a much more comprehensive picture of these microbial communities by looking at the DNA and seeing everything that's present. So this has really been an amazing opportunity to use some of these new methods to study the indoor microbial exposures. Next slide. So there's several DNA-based methods available. The two I'm going to highlight here are qPCR, which is called quantitative polymerase chain reaction. It's a long way to say that you can measure a specific type of DNA that's available in a quantitative way. And all of these different measurement methods have different pros and cons. So at this table here, you can see three different methods that you might use to measure environmental fungi, and across the top are different parameters that you might be interested in for those communities. So culture-based methods are the only way that you're going to get a good indication of viability, but it has limited ability for taxonomic identification and quantification of those microbes, and you can't really use it to measure diversity very well of those communities. But qPCR, which is a way to measure a specific type of DNA that's present, but you have to select in advance. So you design a specific qPCR assay to look for a specific microbe or group of microbes. And that is currently the gold standard for taxonomic identification and quantification, but it can't tell you anything about viability or diversity. And then a lot of the next generation DNA sequencing tools, they can't tell you about viability, but they do a good job with taxonomic identification, quantification if you combine it with qPCR, and then diversity measurement. It's really the best way to get a measurement of diversity. So a lot of what you'll be seeing today are some analyses that we've done that are largely based in using qPCR and these next generation DNA sequencing technologies to study these indoor microbial exposures. Next slide. So how do these analysis techniques work? So if you want to use next generation DNA sequencing, you can your protocol would look something like this. So you take some dust, if you're interested in the exposure from dust, you'd extract the fungal DNA, and then you send it for DNA sequencing, which is a process that usually involves producing amplicons, cleaning, normalizing, and pooling the DNA, and running it on a sequencing machine. From that, you get gigabytes of data, so you get these huge files of sequencing reads. And one of the most challenging part of the process right now is the bioinformatic analysis to take all of those these DNA reads and turn them into identification so that you can get taxonomic identification, quantification, and diversity measurement for the microbes that are present in your sample. But once you are able to do that bioinformatics analysis, you can get hundreds of thousands of DNA reads per sample and get these amazing comprehensive pictures of the microbial communities that are present in any given environment. So these tools right now are primarily being used by researchers to study a lot of these questions, but I see them potentially in the future being more accessible for practitioners to use once we get a more streamlined process going for both the sample collection, the analysis, and then also largely for the bioinformatics analysis. But once that does become more streamlined, it's possible that these types of techniques could be more accessible to practitioners who might be interested. But right now it would be pretty challenging. Next slide. So a lot of this goes really into the long-term goal of understanding how a lot of these exposures are eventually linked to disease. So I'm really interested in the home microbiome, how housing characteristics can influence that microbiome, and how all of that influences our exposures, our own microbiome, and disease, and in this case, particularly asthma. Next slide. So this is kind of the system that I usually think about when I think about these microbial exposures. And I think about how housing characteristics are contributing to these microbial exposures, the microbial measurement, which I was just talking a lot about today, the different processes that are happening in the indoor microbiome, in the indoor environment, and how that eventually leads to changes in human health. So in this section, I'm going to talk about some of the different health studies that we've done linking these exposures to different health outcomes. And I'm also going to talk about some of the microbial processes that are happening in the indoor environment that we're learning more about using some of these techniques. So first I'll talk about the health studies that we've done using some of these measurement methods. So the first question we wanted to ask is what microbes are associated with asthma? There's been a lot of other work done on this. I won't review the literature here, but there's been a lot of great work done in this area, and we were applying some of these new measurement methods to look at some of these associations. And there's kind of two hypotheses out there. I like to think of these as the good guys hypothesis and the bad guys hypothesis. So the hygiene hypothesis is sort of the good guys hypothesis, that there are some beneficial microbes that you need to be exposed to early in life for the proper immune system training, and that's going to make it less likely that you develop allergies and asthma later. And then the bad guys hypothesis is that there are some harmful microbes that if you're exposed to those, they might make it more likely that you develop these things. In reality, in a lot of these microbial communities, it's probably going to end up being a mix of these hypotheses, that actually the vast majority of the microbes there were pretty indifferent to their presence. They're neutral. They don't really do much. And then we're going to have a subset of good and a subset of bad. But these were some of the hypotheses that we were thinking about when we started out with these studies. Next slide. So the first study that we did, this was done pretty early on when these methods first became available. So this was an asthma development study using the CHAMACOS birth cohort out of UC Berkeley, and was also collaboration with the California Department of Public Health. And in this study, we had dust collected at age 1 from homes of these children, and asthma was diagnosed by age 7. And we measured the fungal communities in the dust using some of these new next-generation sequencing technologies. This was a nested case control study where we used all 13 asthma cases available from 292 children, as well as 28 gender-matched controls. And it was based out of Salinas, California. So we were really trying to ask the question, for these early life microbial exposures, what was important for later development of asthma. Also a side note, so this was a cohort of primarily Mexican descent, so the asthma prevalence rates were quite low, which is typical of a population of this background. All right, next slide. So the first result that we got from this was we did not find any taxa that were positively associated with asthma development. So we didn't find any of those detrimental taxa. This slide shows a principal coordinate plot, and in this type of plot, similar microbial communities cluster together. So what we were looking for is a cluster of purple asthma cases and a cluster of gray controls, and that would have indicated that the microbial communities in these homes were very different. And trust me when I say I've spent a long time looking at this plot, but there is no statistical difference between the microbial communities in these asthma cases and control homes. We also analyzed specifically the individual taxa, and we didn't find any that were positively associated with asthma development. Next slide. But what we did find is that we found that low fungal diversity was associated with increased risk of asthma development. So this kind of potentially goes along the lines of having some sort of immune system training that's necessary early in life. So the plots at the bottom, you can see a rarefaction curve on the left and the data summarized on the right with diversity on the y-axis and controls in asthma cases on the x-axis. And you can see that overall the diversity was lower in the homes of children who went on to develop asthma compared to the control cases. And our odds ratio is quite high, it was 4.8. We only had 13 children in this study, so it was a very small study. You can see there's a wide range, but we did get a statistically significant odds ratio that found that lower fungal diversity exposure early in life was potentially harmful and associated with an increased risk of asthma development. Next slide. And the interesting thing is that this association has now also been shown in other studies as well. So here are four birth cohorts in which the same association was found. Here we've taken the inverse of the odds ratio, so high diversity associated with lower asthma risk. You just have to flip it for a minute, but you could see that all these studies are finding this consistent association. And it's really interesting because we're seeing that this low diversity is potentially harmful, but this is also a pretty non-specific measure. So what this measure tells you is that there's a lower total number of fungal species that are associated with asthma development, but we don't actually know what those are, which ones are potentially important. It just seems to be this measurement with the community. And so we keep seeing this association over and over again, but we still don't completely know what it means, and we need to continue to look into this a little bit further to identify what's going on. And there's been a lot of great work looking at potentially what can be going on there. Next slide. Another interesting finding of the study was we identified diversity within a specific genus called Cryptococcus that was associated with asthma development. So this is a fungal genus. Actually, since this publication, a lot of the species within this genus have been renamed, but this is a genus of yeast. And we found that this was particularly interesting because it's also been shown in a rat model as well as an epidemiological study that exposure to a pathogen within this genus might be associated with asthma development. So it's actually possible. A lot of children will develop a subclinical infection with Cryptococcus. This is a chronic infection that oftentimes might be confused for a viral infection because it doesn't respond to antibiotics. But this can be associated with asthma development. And we see this association here with diversity within this genus, which is really interesting because this genus in particular was very difficult to culture. It largely wasn't studied in a lot of these environmental studies because it was so difficult to do with culture-based methods. But now we can see these associations using these new sequencing technologies. Next slide. So for our asthma development study, we saw that low fungal diversity was associated with increased risk of asthma development, and we found some interesting associations with Cryptococcus as well. Next slide. So we also wanted to do a study looking at asthma severity. So for this study, we wanted to ask the question, so for children who already have asthma, how might their microbial exposures be associated with increased asthma severity? So this was using the STAR birth cohort from Connecticut and Massachusetts. It was a prospective study of asthma severity in 196 children. Next slide. So for this study, we broadly divided the children into two asthma subtypes. Sometimes I like to ask different people how many subtypes asthma has, and I feel like I get several different answers, which is interesting. So here we broadly divided them into two categories, allergic and non-allergic asthma. So allergic asthma is mediated by eosinophils and involves IgE, and it's kind of more of an allergic type response. It's what people more traditionally thought of as asthma. Non-allergic asthma is estimated to be the case in about 50% of asthma cases, but was recognized after allergic asthma. And it is mediated by neutrophils and is more of an acute inflammatory response. You could think of it kind of like an irritant type response. Next slide. So we divided the children into this study into allergic and non-allergic asthma. You can see that on the left side of this table here. And then we measured a lot of the different parameters of the fungal communities that these children were exposed to. And we measured the different types of fungal communities as well as the fungal concentration, or the total amount of fungi in their dust. And what we found was really interesting. We found that for the allergic children, it mattered what they were exposed to. So we found statistically significant associations with the fungal communities for their exposure, but not with total amount. So for them, it was more important if they were exposed to things like allergenic fungal species, things like that, or versus how much they were actually exposed to. For the non-allergic kids, it didn't matter what they were exposed to. We didn't find any statistically significant association with the fungal communities, but we found that fungal concentration was important. So the more total fungi that they were exposed to in their dust, the more likely they were to have more severe asthma. And so this kind of makes sense that for the allergic children, it matters what they're exposed to, and for the non-allergic children, it matters how much, which is consistent with that irritant type response that they might be having. Next slide. I won't go through all this table, but we did look at the specific taxa that were associated with asthma severity. And the interesting thing here is that while not all of these were statistically significant when we controlled for multiple comparisons, we see a lot of Cryptococcus showing up as sort of marginally significant. So we see the same genus of yeast as potentially being important for asthma severity, and we need to look into that a little bit more. We also identified Candida yeast as potentially protective, and that was also potentially protective in our asthma development study. So it's interesting to see another yeast showing up. Next slide. So overall, we found that asthma development and severity were associated with different microbial exposures. So for asthma development, we found that overall microbial diversity was important, and asthma severity was children with allergy, it mattered what they were exposed to, and children without allergy, it mattered how much. So these new DNA-based microbial measurement technologies were really able to allow us to find a lot of these new and exciting associations between exposures and asthma. Next slide. And we also found some really interesting associations with yeasts, especially Cryptococcus and Candida, and so that I think is a really interesting area for future research. Next slide. Okay, so I just talked about the section of different health associations with a lot of these microbial exposures. So another area of research that we're interested in is not only if these exposures are associated with health outcomes, but also mechanistically what's happening in the indoor environment. And we know that microbial exposures are important for these health associations, and we also know that moisture is harmful. So we know that having moisture and dampness problems in a building is potentially harmful for some of these health effects. But we wanted to ask the question, what's going on when there's a lot of moisture in a building? And specifically, we wanted to look at carpet because you can get high resuspension rates from carpet, and ask, what happens if you have elevated relative humidity in a room? Is that sufficient to support microbial growth in carpet? Next slide. We are really interested in carpet because it's so prevalent in homes. So you could see that the carpet here's sort of an estimation of carpet versus non-carpeted floors, and you can see that it probably is present in about 50% of the rooms that were measured. And it's such an important flooring material, so we wanted to specifically study what was going on there. Next slide. And we also studied carpet because we know that the relative humidity in carpet can be actually higher in a room than what's present in the room air. So if you measure the relative humidity in the middle of the room, it could potentially be lower than what's present in the carpet, depending on the room itself and history. And there's a lot of different things that could be happening in that carpet, but we wanted to focus specifically on the fungal communities and what could happen to them if the relative humidity were to increase. So in this section, I want to talk a little bit about microbial growth in carpet. We collected carpet and dust from a home. We exposed it to elevated relative humidity conditions. So we considered elevated relative humidity to be above about 80%, which is pretty high for a house, but not outside of reason. So that could definitely happen in a bathroom with someone showering. If you did have some damage like from a water leak or something like that, or possibly in other situations such as maybe after a cooking event or something like that. Also, maybe depending on climate, if you're in a hot, humid climate, you have the windows open, relative humidity could go up. And then after simulating those relative humidity conditions, we wanted to measure microbial growth, so we looked at rate, function, and then eventually we also looked at their interaction with some chemicals in the environment. Next slide. So to do this, we collected carpet from a home. In our method, we go and sample a carpet. We were looking for a carpet that was going to be thrown away for aesthetic reasons, so there was no water damage in the home, anything like that. We applied some dust from the same house to the carpet, and we embed it using a modified ASTM method, and then incubate it under temperature and relative humidity controlled conditions. And so then we wanted to ask, are we going to get microbial growth in this carpet? Next slide. So our first indication that this is what was going to happen was when we looked at the water activity of the dust. And water activity is a measure of water availability. You can think of it as in equilibrium with the relative humidity. So if your relative humidity is 80%, the water activity at equilibrium with a solid or another substance is going to be 0.8. And we saw that the relative humidity and the water activity of the dust correlated very well. You can see some pictures of the dust here. This is just the dust, not embedded in carpet, after 1 day and 1 week at different relative humidity levels. And you could see a visual color change in the dust, and we knew we were onto something when we looked at the 1 week 100% and saw visible fungal growth on the dust itself. Next slide. So indeed, microbial growth can occur in dust at elevated relative humidity levels. So in this slide, you could see we measured the spore equivalents per milligram dust for fungi on the y-axis, or the amount of bacteria on the y-axis, and the original dust is the OD, or the percent equilibrium relative humidity that it was incubated at. And so you can get the different concentration levels. And you could see that as the relative humidity increases, we get more fungal concentration, starting at about 80% relative humidity. For bacteria, we see an increase only at 100% relative humidity, and not quite as large of an increase as the fungi. One funny story about this is when I started this project, I actually wanted it to be a bacterial-based project, but the story is actually all with the fungi, so I should have seen that coming. But at the bottom two graphs, you could see that this continues over time at 85 and 100% relative humidity. The samples at 100% relative humidity, we were also planning to continue out to 6 weeks, but they became so covered in fungal growth that we decided to stop that experiment, and that we had seen that it does indeed continue. Next slide. So we saw that fungal growth can occur in dust at elevated relative humidity levels embedded in carpet. We wanted to measure the specific species that were present. And a lot of the species listed here that we identified are those that are known to be associated with water-damaged buildings. So a lot of Aspergillus and Penicillium type species that are known to be measured commonly in water-damaged buildings were also found in our dust that was elevated at elevated relative humidity levels. Next slide. And we found that this could be a potentially important source for human exposure. So if you look at these graphs in orange, we have the background exposure that could occur under regular conditions. And then if dust was at these elevated relative humidity conditions for 1 week, you could see the additional microbial exposure that you might experience. And this is kind of an odd laboratory conditions. I definitely wouldn't recommend anybody to leave their house at 100% relative humidity for a week. But you could think about it that if these elevated conditions were extended out over time, how you could end up with some really problematic exposures, especially if you have some of these species growing that contain known allergens. So this can definitely the growth in the dust can definitely have an important impact on human exposure, depending on the relative humidity levels in the house. Next slide. So another thing that we wanted to look at this next was actually what's going on in these microbial communities. So we wanted to look at microbial function and basically ask the question, what are they doing? And so we looked at the gene expression of these communities. We actually measured the RNA, which is tells us if these genes are being expressed. And the interesting thing that we found is that we did samples at 50%, 85%, and 100% relative humidity. And we found that the 50% and 85% samples were actually very similar to each other in terms of the genes that these microbes were expressing. But once you get very high moisture levels, around 100% relative humidity, you get a very high level of diversity in terms of what these microbes are doing. So if you look at the principal component plot here on the left, the red samples are the ones incubated at 100% relative humidity, and they're different not only from the 50% and the 85%, but also from each other. And you can see that on the heat map too. Next slide. And you could see this, I don't know if the video is going to show up. It is actually a GIF, but I can just describe it. At 50% relative humidity, you have a lot of these core metabolic processes occurring. So these spores, they don't have a lot of moisture available. They're just barely hanging on to life. The lipid metabolism and carbohydrate metabolism, things like that are complete, but that's really all that they're doing. You get a little bit more activity at 85%, but at 100% relative humidity, you see all of these secondary metabolic processes light up in these spores. And the interesting thing about that is that a lot of allergens and virulence factors and mycotoxins tend to be present in these secondary metabolic pathways for these fungi. So this means that when you have increased moisture available to these microbial communities, you actually potentially get a lot more of these allergens and things like that. Next slide. And so we wanted to test this. So we actually looked at associations with these humidity levels and allergens, and we found many more allergens were actually expressed at 100% relative humidity levels. And we also tested this with a protein-based assay to measure the allergenicity of a single fungal species, and we found that increased water availability was actually associated with increased allergen levels. So this kind of begs the question after the previous analyses is a spore? Or if you're exposed to a fungal spore elevated at these elevated relative humidity levels, is it much worse than being exposed to another spore grown at lower levels that maybe doesn't contain as many allergens or other harmful factors that you might be exposed to? I see a question. I might go ahead and answer this question real quick. So the question is, in regards to recommendations to measure relative humidity in homes, several of us have worked on or are working on research protocols that involve using sensors in the homes of people with asthma to measure relative humidity. Do you have a recommendation for sensors and or placement in the home to measure? And any additional considerations when the room is carpeted versus not carpeted from your perspective. I do think it's really important to maintain the relative humidity levels, especially as indicated by the EPA, which are that homes should be maintained between 30 and 50% relative humidity and definitely below 60% relative humidity. I think it's okay to measure relative humidity where you can in a room, and but think a little bit about any cold surfaces. So it's oftentimes a problem if you have a carpet that's attached to like a concrete floor that's going to be a lot colder than the rest of the room. You might want to think about having some extra humidity control in a situation like that, or ideally remediating that situation. But cold surfaces can definitely have a higher relative humidity level on them compared to what's at the center of the room. And in terms of specific devices to measure relative humidity in a room, I think you can get fairly accurate meters that are pretty widely available. So there's not a specific recommendation that I have for that. But thank you for the question. All right, so the last section of my talk that I wanted to talk about today is also what these microbes are doing. So we saw before that these microbes have function, especially if relative humidity level is high enough in a home. And so we wanted to look at what they're doing. And so I was able to bring this back to my chemical engineering background and say, are there any interactions with chemicals in the home? And so we wanted to look specifically at phthalates. So phthalates are important because they're plasticizers, but they're not covalently bound to plastic, so they tend to leach out in the indoor environment. They're found in elevated levels in dust, up to 1 milligram per gram in very high levels. That's a particularly high case, but they can be found in fairly high levels. They're of public health importance because they're endocrine disruptors. And they can be degraded by microbes in other systems. So we wanted to ask the question: do you think these microbes could be degrading phthalates in these environments as well? Next slide. So we incubated dust at elevated relative humidity conditions and measured phthalates in the homes. And we did 2 experiments. We had a natural experiment where we measured 9 naturally occurring phthalates, and also a spiked experiment where we spiked deuterated phthalate into the dust and measured its degradation. Next slide. And I'll skip this. Next slide. So one thing that we found when we incubated these carpet samples with these phthalates is that we looked at the 9 phthalates that were present in the dust. And we noticed, so on these graphs you have time on the x-axis and the concentration on the y-axis. And we saw degradation of high molecular weight phthalates, but not so much the low molecular weight phthalates that were present. So we saw that we had this degradation of high molecular weight phthalates at these elevated relative humidity conditions. We thought that this was really interesting. At this point, we couldn't say if it was chemical or microbial processes, but we wanted to look into this further. So we selected one of the high molecular weight phthalates, the DEHP, for our spiking experiment to look at microbial degradation. Next slide. And indeed, we found that microbes are capable of degrading phthalates in house dust at sufficiently high moisture levels in these homes. So we saw, we did an experiment in such a way that we could differentiate between chemical degradation and microbial degradation, and we found that both processes were important for degrading phthalates in house dust at these elevated relative humidity conditions. So the microbes in these homes could actually interact with the chemicals in the dust and cause degradation of these compounds. And you might think that this is potentially beneficial, but actually a lot of these degradation products are potentially even more harmful than the parent compounds could be in these homes. Next slide. So we looked at some of the degradation products as well, and we found that this was consistent with the reduction in the parent compound. So we did see the DEHP was being degraded to MEHP, a monophthalate, as well as pentophthalic acid, which both potentially have harmful effects. So all 3 of these compounds could potentially have harmful effects. Next slide. And it was interesting because in a lot of other systems, we see that microbes preferentially degrade low molecular weight phthalates, but here we saw that they degraded the high molecular weight phthalates preferentially. But we were actually able to look at the gene expression and the function of what these microbes were doing. And with this type of analysis, we weren't able to definitively identify which degradation processes were occurring, but the ones that we saw suggestive evidence for indicated that these microbes might preferentially degrade the high molecular weight phthalates. Next slide. So overall, we found that microbes could interact with these chemicals in the home, and we were really excited to find the potential for this interaction. So to summarize the microbial parts of my talk, indoor fungal exposures are really important for human health. So we saw associations with health outcomes, and we need to continue to learn more about those using a lot of these new measurement methods. We found that elevated moisture was sufficient to support microbial growth and function in dust, and so we're just beginning to understand what these microbes might be doing in our dust if they have sufficient moisture available. And third, we found that this has implications for many environments, both on Earth and beyond. So I'm really excited actually to be looking at some new samples from the International Space Station to understand what might be going on in dust from that very specialized environment and look at the microbes there. But I think this has really important applications in a lot of areas. Next slide. So overall, once again, the idea of this work was to look at, use these new measurement methods to link these environmental exposures to health outcomes. Next slide. So I have a lot of people to thank for this work in part two. There's a lot of different funding agencies. The Alfred P. Sloan Foundation funded a lot of this work, as well as NSF, NIH, EPA, and the Guizetta Foundation. And there are a lot of different people involved in this work and the various papers that have come out of this. So I definitely couldn't do it without them, and I'm really thankful to be working with them. Next slide. All right. So thank you very much for having me. It was great being here. If there's any time, I'd love to take some questions.
Yes, thank you so much, Karen. This is really. Can you hear me?
Yes, I can hear you.
Yeah, thank you. This is really good. I am really so amazed how you have connected different types of disciplines together and make this research going on. I really enjoyed it. Any questions from our audience?
I see one here. So it says, you mentioned formaldehyde is ubiquitous. However, are there high-risk environments or regions that should be tested, or is testing usually done to determine source of already present health problems? That's a great question. So asking if there are high-risk regions that you should be concerned about. I think that a lot of people might potentially be testing because they have some health concerns that they're worried might be linked to formaldehyde. New buildings are known to have higher formaldehyde levels because they have a lot of newer materials that are still off-gassing formaldehyde from a lot of those materials. So that's somewhere that you could consider looking at. In terms of the best thing to do is to try to reduce the use of these materials that might tend to off-gas or select materials that maybe have lower formaldehyde levels. You could also think of other areas of concerns or maybe hobby areas where people are using a lot of adhesives or paints or things like that. Those are some areas that might be of concern. But great question.
Mm-hmm. Very interesting.
Any other questions? Or right, I think that's it. I don't see any new questions here. Thank you so much, Karen. We appreciate your time, and if you have any new research going on, any new publication, please let us know. We may invite you again to let us know what you're doing.
Great. Thank you so much for having me. I had a great time being here. And if anybody has any more questions, feel free to reach out. My email is up there. My Twitter is up there. So definitely let me know if you have any questions.
Thanks so
Thank
much.
you. Thanks, everyone. Bye-bye.
Thank you.